Hello everyone, and welcome to today's live webinar broadcast hosted by Organovo and presented by Dr. Ben Shepard, Director of Therapeutics at Organovo. My name is Tom Murphy. I'm the Director of Scientific Applications here at Organovo. Before we get started, I would like to remind everyone in the audience that we are broadcasting live, and following the presentation there will be a Q&A session offering you a chance to pose questions to our panelists. We encourage you to submit your questions at any time throughout the presentation by typing them into the question pane of the control panel. We will collect the questions and address as many as we have time for following the presentation. And with that, I'd like to introduce Dr. Ben Shepard. Ben has over 17 years of experience in tissue engineering and regenerative medicine research. Dr. Shepard's research in vascular biology and microvascular tissue engineering has been focused on the ability to use creative approaches to generate microvascular networks to support the fabrication and surgical implantation of prevascularized neotissues. Prior to joining Organovo, he was Associate Research Scientist at the Yale School of Medicine, where he also conducted postdoctoral training. In addition to his appointment at Yale, he was a staff perfusionist at the University of Connecticut Health Center. Dr. Shepard is a board-certified clinical perfusionist, and he received his PhD in physiological sciences from the University of Arizona and his BS in zoology from the University of Washington. And with that, I'd like to turn it over now to Dr. Ben Shepard.
Thanks for the intro, Tom, and good morning to everyone who's joining us online. So I'm going to walk people through a little bit of our transition from development of in vitro tissues that have been useful in kind of the preclinical space and in the laboratory setting to efforts to develop and fabricate bioprinted tissues in the clinical space that would be useful for treating patients, and specifically patients that are suffering from end-stage liver diseases. So before we get started, I'd just like to remind people that as a publicly traded company, I'm likely to make some forward-looking statements today, and I would encourage anyone who's interested in finding the relevant information on our financials and regulatory filings to go to our website where they can find that information, including our most recent Q filing. So briefly, just as an overview and an outline of what we're going to talk about today, I'll kind of walk people through a little bit of the basics of the Organovo bioprinting platform. That is, what is bioprinting? How is it that we capture some of the useful aspects of nature and exploit that to make tissues and fabricate three-dimensional biological systems? And then give a case study about how one might think about using those in the laboratory, how they might be useful, and where that impact may be. And then kind of translate to the development of therapeutic tissues. And essentially what we would like to do is to take a lot of our learnings from the in vitro space and from the lab to more well inform our development of therapeutic tissues and think about how we can understand a little bit of the behavior of those systems in order to have a more significant and perhaps more rapid impact in the clinic. So I think that we would probably consider ourselves a three-dimensional biology company as much as anything. So we are absolutely interested in understanding how cells and tissues behave when placed in some sort of a three-dimensional arrangement that is more tissue-like than you would find in traditional sort of laboratory 2D cell culture systems. And certainly we use bioprinting as a way to get to that end. And so we're able then to take largely human cells and combine them in specific patterns in some automated, spatially controlled deposition system. And that allows us then to have a reproducible fabrication strategy where we can many times over have replicate tissues across, in this case, as you see in this image here, across a plate, for example. And importantly, the finished product, the finished tissue, is absent the traditional sort of degradable polymeric scaffold materials that are often found in the classic tissue engineering approaches. And in many instances, those scaffold materials have been found to be inflammatory and perhaps compromise the overall function and performance of those tissues. And so we're left with a tissue that's comprised really just of the relevant tissue cell type specific materials, and then importantly, the extracellular matrix proteins that those cells produce, and that then yields some sort of biomechanical integrity and the important biomechanical cues that one would expect to find in a naturally occurring tissues. And so, this is a slide just to kind of illustrate the basic workflow of how we do that. So we work primarily with human cells. I would say probably 95% of the R&D work that's been conducted, both at Organovo and kind of the foundational work that led to the foundation of the company, that's all been done with human cells. And by and large, those are primary human cells that we procure from donor tissues, cadaveric donor tissues, but can also include stem cell derived populations, whether those are from induced pluripotent stem cells or embryonic stem cells. The system is quite agnostic to what those inputs are, so it allows us the opportunity to evaluate those materials as well. And we expand those cells under kind of standard classic cell culture conditions on plastic or on some sort of microcarrier system, whatever is most suitable for the end goal. And those are expanded up to sufficient numbers and then prepared as bioink. And importantly, the bioink that is generated from those cells can be created in a number of different ways. And specifically what I'm thinking about is different combinations of cells, different proportions of cells, and perhaps different materials into which you prepare that. So different excipient materials that will facilitate the actual printing process. And then from there, after the generation of that bioink, those materials can then be loaded onto the bioprinter itself. And in this case, this is an example of a four-headed bioprinter, and one can then understand, I think quite easily, how altering what those bioink compositions are, and then the series in which those are deposited, you can start to increase the geometric complexity of what it is that you are generating, what it is that you are fabricating. And in this image here and in this workflow, what we've done is then print a plate of tissues. And in this case, perhaps these are liver tissues that we would use in our commercial offering, which we'll talk a bit about here in a few minutes. And what you have then is a plate really of replicate tissues. That is, all of these tissues have the same fundamental building blocks to begin with, and they can then be interrogated in the laboratory with some specific stimuli to understand a little bit of the response across a plate of common starting points, and then we can evaluate how they may respond to different stimuli with some specific readouts, and that may be conditioned media, that is protein or enzyme production. We can look at kind of the classic omics, that is, can you isolate protein, can you isolate RNA or DNA, and understand what those differential responses are like. And then importantly, we have some mass of tissue, and we can put that through kind of a standard histopathologic or surgical pathology evaluation to understand how the different cells are behaving and what type of tissue pathology might be developing. And so then you might ask, okay, so how is it that you're able to do that? And this slide is meant to illustrate sort of one of the key components of how you go from isolated single cell on plastic and then suspension to kind of aggregates or clusters of cells that form some sort of a tissue-like structure. And so the cluster of images on the left, this panel here, shows what an aggregate of cells would look like, and in this case, two aggregates of cells when placed in close proximity to one another over the course of time. If placed close enough so that they essentially just touch, they will fuse, and in this case, over about 1400 minutes to form a single structure. And one can then think about making a geometric structure by arranging those aggregates of cells in a pattern, allowing them to fuse over the course of a few days into, from originally kind of this ring in what looks almost like a dot matrix pattern, to this single continuous tubular structure. And so in this example, this is over the course of maybe 120 hours, but in reality and practical purposes, this process occurs quite quickly. And so we're able to take advantage of that phenomenon many, many times over within the tissue to go from sort of a loose collection of aggregates of cells ultimately to a cohesive tissue structure and how we go from the initial starting material to that end material is really sort of where all of the magic happens, so to speak, and where most of our intellectual property lies, whether that's in actual specific IP filings around patents and disclosures, or trade secrets. That is, how one prepares the cells, how one prepares the bioink, and then how, after you've fabricated a tissue, you might mature it then into your ultimate goal, which it would be a native tissue-like structure that one could interrogate. And this slide really highlights what we've been able to do as a company in terms of collaboration. So we've had very successful, very fruitful collaborations across a broad array of partners that are representative of both the large pharma space as well as the academic space. So and additionally some institute partners such as the NEI, National Eye Institute, and NCATS at NIH. And by and large, those partnerships are meant to expand and complement the research activities that occur at Organovo internally. And so we might look then to partner outside of, for example, the liver space, and perhaps work with someone that's doing some sort of eye work. Maybe it's vascular biology or oncology. But by and large, we look to give access to the platform and to our internal capability to people, really smart people in other labs, that they can play with the system, they can develop their own IP, and then by doing that, we can certainly expand what the capacity and the bandwidth is of the investigators here at Organovo. And so I'd like to spend a couple of minutes and walk people through what we envisioned the tissues would be useful for at the outset, and that is to expand the ability of drug scientists to identify new drug entities that may in fact have a tox signature or have some sort of biological response that wasn't identified with the currently available systems. That is, currently available cell culture systems or perhaps animal systems. And so I'd like to walk through a case study of how that might be actually utilized. So many of you are probably more familiar with our commercial offering, our in vitro or non-clinical work than the things we'll talk about later, which is some of our newer data in the clinical space and the therapeutic space. But just to orient people who are not previously familiar with that, the ExVive liver tissue contains fundamentally 3 primary cell types. So those are human hepatocytes as essentially the parenchymal cell type in this tissue, and supporting endothelial and stellate cells, which are important mediators both of natural biology and natural physiologic responses, but also of pathologic responses to external stimuli. In some cases, for some investigators, where they may be interested in some more specific biology, maybe perhaps inflammatory biology, or a specific external stimuli, we can also incorporate Kupffer cells, the resident macrophage or reticuloendothelial cell of the liver. So that is an additional possibility. So those cells are captured and are prepared in bioink, just as we talked about in the first couple of slides, to generate a tissue, as you see here. And that tissue is generated and fabricated directly on a transwell membrane, which is important because it allows us then to have media access essentially surrounding that developing tissue. That allows for proper maturation, but then also allows for stimulus at the level of the entire tissue. So one can place the xenobiotic or the new drug entity or whatever compound they're interested in investigating directly into the system, get exposure to the entire tissue, and then you can harvest that tissue and all of the conditioned media and do the assays that we talked about before. So whether that may be evaluating protein production and content of the media or the tissue, we can also look at RNA expression profiles. And then importantly, because we have fabricated a tissue-like structure, we can start to actually look at what the tissue looks like at that cellular and organizational level. So here in this image, these dashed lines highlight a collection of supporting non-parenchymal cells, which, as we described earlier, are highly enriched for endothelial and hepatic stellate cells, and those are surrounded by these hepatocyte-rich parenchymal zones. And so the first thing that one might do is to just kind of characterize a little bit of what that tissue looks like at baseline in the absence of any stimulus. Does it in fact have hallmark characteristics of liver? And so we could look for important adhesion molecules like E-cadherin. Perhaps the largest, most kind of voluminous protein produced by the liver is albumin. So these hepatocytes you see in red are full of human albumin. We can then look at the non-parenchymal cell organization. So the red staining here in this middle panel, is human CD31. This marks the endothelial cells, the cells of the vasculature. And so you can see them luminizing and organizing. And as one would expect, both between hepatocytes and along the developing vessel wall, you find supporting hepatic stellate cells. We can also do EM. So in this case, this electron micrograph, you can clearly see the development of bile canaliculi as well as the tight junction and adherens junction formation. We can look to see, does it store, does it stain positive for lipid? So in this case here, this image is stained oil red O, and you can see bright red lipid droplets in the liver as one would expect. We can demonstrate glycogen storage with periodic acid shift staining. So this very nice staining pattern here shows, in fact, that the hepatocytes are full of glycogen and doing hepatocyte-like things. So taken together, there's a nice description of what the tissues look like at baseline. They are organizing themselves into tissue-like structures and doing the things that you would expect of hepatocytes in liver. And so the question then is, obvious is, okay, that's fantastic. What would you do with this system? And so we went back to the literature and pulled a very nice example of a drug that made it through all of the available testing mechanisms that were available at the time. Essentially, in some countries to market, in others very, very late clinical trials, before liver toxicities in patients were observed. And so this is, a significant setback in a number of different ways, not the least of which is the impact on patients, right? So there were a number of severe liver reactions, including, in addition to acute liver failure, there were a limited number of deaths. And it's a very significant cost burden to the development cycle for that company in terms of their R&D pipeline. So the concept here would be then, can we take replicate tissues, as you see here on the left, expose them over some period of time to a varying concentration, so a dose response experiment, if you will, ranging from the Cmax, that is the maximum expected dose in the clinic, above and below that, so half and about 20 times that, and then look at some specific endpoints. So ATP as a marker of overall cell viability in the tissue, albumin production as a marker of specifically hepatocyte health and their ability to produce protein. And then we can, as we touched on before, actually look at the development of the histopathologic response to these tissues, to this compound, excuse me. And importantly, in the case of trovafloxacin, there is a structurally related compound, levofloxacin, which is safe. So this is on the same pharmaceutical background and scaffold with a slight modification. And what we know now over the course of many years of patient administration is that levofloxacin is quite safe. And so if we were then to look here at this bar graph on the left, this is in blue levofloxacin dosing. And until you get much, much above the Cmax, you have very nice sustained production of albumin that is essentially the same, not statistically different, than the media-treated control tissues. If you contrast that with the trovafloxacin-treated group, even below that clinical max, so well below that, about 0.5 of the Cmax, you see a quick and significant reduction in the amount of protein that is produced, indicative of broad liver injury. And if you were then to look at ATP, so this is essentially indicative of the entire tissue, while you maintain ATP production that is essentially the same as vehicle. Once you get above the Cmax in the case of trovafloxacin treatment, you begin to see a significant reduction in ATP that really highlights the loss of viability across the tissue. And on the right here, if we were to look at what those tissues look like, in this case by just simple H&E staining, what you see on the left, above and below, are the media-treated control tissues, and on the right, what you see are the trovafloxacin-treated. And what at low power there across the top is quite easy to see, is essentially a dissociation and a loss of adhesion within the tissue in response to treatment. And when we look at the core of that tissue, we can see evidence of clear hepatocellular necrosis that looks quite reminiscent of coagulative necrosis in these tissues. So importantly, we were able to demonstrate this phenotype in the absence of any exogenous stimulus with something like lipopolysaccharide. So LPS has been used in other systems to generate a similar phenotype, but prior to this data had not been widely reported. It had not rather been widely reported that you could observe this degree of trovafloxacin toxicity in the absence of LPS stimulation. So it suggests to us that this is the fidelity of this system is quite nice, and it's a good example of how one might have been able to tease out and identify a potential toxicant long before it had made it into clinical trials and patients were exposed to a harmful compound. So the idea really here at Organovo has been to understand as much as we can about building those tissues, the behavior of the cells, the behavior of the tissues, in terms of both the fabrication and the maturation and what that expected biology profile might look like, and to use that data and to use that experience to develop therapeutic tissues. So ultimately, we certainly would like to have an impact in both the preclinical and the clinical space, and we've begun to make a very concerted effort at doing that second half. So I think that this slide is probably not unfamiliar to anyone who's tuning in today. The bar graph here, or excuse me, the line graph there on the right demonstrates in gray the number of patients on the transplant waiting list in the U.S. That number has continued to increase, is expected to continue to increase as the patient population ages, and as we start to grapple with a little bit more significant disease states, specifically in the liver and the kidney space. And interestingly, the number of transplants conducted may have risen slightly, but the number of donors available has actually begun to decline a bit. So the gap between really the available tissues for transplantation and those patients requiring or needing those tissues has widened significantly in recent years, and that widening is expected to continue for some time. So, people have made efforts at trying to do direct transplantation of allogeneic hepatocytes, and there have been some encouraging early results, but there are some persistent challenges. And so the delivery of kind of a homogeneous cell suspension is difficult for a couple of reasons. So the precise delivery of cells is difficult in the absence of kind of a direct injection. Even in the setting of a direct administration of kind of a suspension or even a concentrated suspension of cells, there's a fairly rapid clearance by the immune system and inflammatory biology that occurs. And as a result, engraftment of those cells is quite variable and typically very, very low. And so people have tried some extracorporeal device systems, but those have really failed to demonstrate clinical benefit and efficacy in recent trials. And it seems that perhaps that's due to the sourcing and the viability, perhaps the variability of the cell material that is used in those systems, but may also just reflect the fact that it's an extracorporeal device, and you're not really administering the cells to the site where they're most needed, and they would naturally occur. And so, taking all of those things into account, we really have some specific design criteria in mind, right? So we obviously would like to use our own in vitro biology platform as the point from which to launch these efforts. The construct and the tissues we think needs to have a very high cell density, and that, as we've seen in our commercial offering, the ExVive liver tissue system, we would like to see tissue-like native architecture that results in an increased functionality. And we are hypothesizing essentially that approach will improve the retention and the long-term engraftment of the target cell type, in this case, hepatocytes. And if we use our fabrication approach, we should be able to limit, if not completely exclude, the inclusion of immunogenic or inflammatory biomaterials. And then importantly, because we allow the tissues to mature for some period of time, we can produce an easy to handle, that is for the transplant surgeon who's potentially conducting these implants, and that we can have this kind of modular design that allows us to scale up and specifically target both the dosing and the fabrication strategies, which will be necessary for scalability and patient dosing. Initially, I'd like to point out our focus is really on an allogeneic approach, so we're not using a stem cell or an autologous approach today. We are focused on using allogeneic donors, with the understanding that these patients are going to be the recipients of transplantation at some point, and we would like to increase the time that they have until transplantation is critical, and we think that that's a significant add to the quality of life for those patients. So this really here is fundamentally at the basic level what we think you would need to see. That is a defined area of non-parenchymal and parenchymal cell population. So you can see the hepatocyte-rich region in this tissue. This is obviously the most fundamental geometry that one could think about, but it meets all of the basic demands for our preliminary experiment. And then here what you're seeing is the tissue sutured onto a piece of muscle in the laboratory, where we can pull on that and assess its biomechanical integrity. And in this image here down below, this is actually a screen grab from a video where the tissue has been rolled up and then rolled back out to make sure that it maintains its geometry and its structure. And there just were initially just some crude assessments of, could we hand this to a microsurgeon at a different laboratory, naive to the program, naive to the system, and could they then manipulate that and implant that into a recipient rodent? And so at this point, we're able to meet that bar quite easily. And so we did a bit of characterization of these tissues. They are slightly different than we have seen in the ExVive commercial system. So their geometries are a little bit different, the proportions are a little bit different, and as we touched on earlier, that's an important part of kind of the trade secret of what it is that we're doing. But we can identify, just as in the other tissues that we talked about, areas that are enriched for supporting endothelial and stellate cells, and then areas that are enriched for hepatocytes. And so this is at that junction between those two components, and this is directly in the middle of where you would find a hepatocyte-rich region. So if one were then to ask about the specific phenotype of those cells, we can identify by human CD31 staining in green, which will highlight the endothelial cells. We can see here, even early on, this is about 3 days post-print, we can see very nice organization of endothelial cells into vessel-like structures, and albumin production within the hepatocytes of the tissue. And so the first question that we really asked was, what would happen if we implanted these tissues after some period of maturation onto the surface of immunocompromised mouse livers? So the image on the upper left, the H&E staining, what you see here at the bottom, this is the implanted bioprinted liver tissue. Above here in the upper left is the recipient mouse. And so these are non-injured immunocompromised control animals. This is 7 days after implantation. The yellow arrows here are calling out the perfused vasculature that is surrounding the tissue. And what you see here on the right is albumin staining. So this is human albumin lighting up and highlighting only the human hepatocytes. And what we're able to clearly delineate here at 7 days is retention of that original geometry. So you can see the hepatocyte-rich zone with infiltrating endothelial cells, and that's surrounded by an endothelial cell-rich and well-perfused area here. You can see it in a slightly different plane. The vascular profiles by human CD31 staining. And we can then further characterize the hepatocytes by staining for factors here, in this case, clotting factors, so human fibrinogen and factor 7, only identified within the human hepatocytes. And we did some further characterization. We wanted to understand a little bit about the distribution of cells, both within the tissue and in the underlying mouse tissue. And what we have found is that the human CD31, human albumin, and really, if we just look for human leukocyte antigen, that is HLA staining, that is largely confined to the tissue itself. We see very, very limited migration of some of the non-parenchymal cells into the very close surrounding mouse tissue. We don't see evidence of distant migration, and we see little or no evidence of migration of the human hepatocytes into the underlying mouse tissue. We're not necessarily certain what that means, if that's good or bad. It tells us, we think definitively, that we have very nice retention of the tissue, and it gives us a good way to look at the long-term development of the tissue, in response to healing, and then ultimately in response to injury, which we'll talk about later. We have also been interested, at least in this non-injured animal model, we've been interested in what the healing looks like over the course of at least 28 days in these experiments. And so we evaluated some animals over that time course, and then looked for some additional markers in addition to the clotting cascade. We were looking at, in this case, ornithine transcarbamylase, OTC, and FAH. These are key enzymes, key metabolic enzymes that are present in hepatocytes, but importantly represent targets, patient targets. That is, patients suffering from inborn errors of metabolism, so OTC, one of the urea cycle deficiencies. Those IEM, or inborn errors of metabolism patients, are likely targets for us. That is to say that those are patients that have limited options today and are prime candidates for cell therapies, regenerative medicine therapies, where one can add back these enzyme deficiencies because a small addition to their deficiency is actually quite clinically relevant. And so, in further characterization of some of these, we've looked within the core of the tissue. We see the development of sinusoid-like vascular structures. So you can appreciate here in this trichrome stain a bit of these, kind of columned erythrocytes here, and you can see development obviously in this blue, which is highlighting collagen staining of ECM between hepatocytes. Again, kind of a hallmark feature of what you would expect in liver parenchyma. And if we were to characterize a little bit further some of these longer-term implants, these 28-day implants, and ask the question about long-term retention. So this collection of images here on the top, across from left to right, is our days 7, 14, and 28, and what we can clearly see is retention of perfused microvasculature in those areas that were enriched for endothelial cells, and retained hepatocytes within the core. And if we look for staining of specific human proteins, and in this case, human albumin, these images across the bottom, these 3 panels, are all from 28-day animals. We see, albumin-rich hepatocytes. We find organized CD31 positive human endothelial cells. And if we were to stain for human leukocyte antigen, we can see that the vast majority of these cells are still human at 28 days. So there clearly are some infiltrating murine cells, specifically at the edges, but the majority of the cells within the implants are still human after 28 days. We have additionally looked at clotting factors, so some of the specific hemophilias are potential targets for us as well. These clotting factor deficiencies are another patient population that may be amenable to treatment, and so we've looked at things like factor 10 and factor 9, and in this case factor 7 and fibrinogen, out as long as 28 days, and can find not only positive staining, but positive staining in a pattern that you would expect. So, for example, factor IX should be kind of in these small vesicles, and in some case in extracellular positions closely associated with hepatocytes, as you see here, while fibrinogen should be found essentially within the hepatocytes in vesicles, as you see here. And so we can also then look at the circulation of these mice to see if the hepatocytes that we have engrafted are functional and communicating with the murine circulation. So in this case, we've looked at the presence of human albumin, human alpha-1 antitrypsin, and human fibrinogen within the murine circulation. And over the course of 28 days, as compared to the sham animals, you see nice production of protein in these recipient animals. So importantly, we believe and are starting to generate evidence that in these non-injured animals, that protein production is evidenced quite quickly, and that the production is maintained for at least 28 days, albeit at kind of this modest state. We're now interested, and I'll start to present a little bit of data to show where we're going with this, but we're interested in understanding now how these tissues respond to injury. So it's well described in the literature that these sort of endogenous regenerative restorative cues in the liver are very, very important, not only for the host liver, for the native responses to injury, but also to promote engraftment in the setting of cell transplantation, or in this case, bioprinted tissue transplantation. And so one of the model systems that we've begun to look at is known kind of as this PyZ mouse. So this is the first data set that we've started to talk about in terms of implantation in this setting of injury. So the PyZ mouse is a transgenic mouse that is overexpressing the mutant form of alpha-1 antitrypsin, known as the Z variant, the PyZ, and as a result, mouse hepatocytes, which are expressing that, become laden and with this cytotoxic alpha-1 antitrypsin. So as that A1AT accumulates in the system, it becomes cytotoxic to the hepatocytes as it gets stuck essentially in ER-bound globules. And so as a representation of what that looks like, if we are then to transplant human hepatocytes in our bioprinted tissue onto the surface of these mouse livers, what we find is very nice homogeneous staining of alpha-1 antitrypsin, that is diffuse cytosolic staining. And if you compare that to the underlying mouse tissue, what you find are large globules of mutant A1AT, this Z variant protein within the hepatocytes. And you can see this hepatocyte here. This is a single, very, very enlarged hypertrophic hepatocyte that is full of A1AT globules. And over time, that hepatocyte will essentially apoptose as a result of that cytotoxic protein accumulation. So the first question that we really wanted to understand was, if we were to implant in this setting of injury, would there be an impact on the degree of protein production, of human protein production in the rodent circulation? And that appears to be true. So what we see now is that for as long as 63 days, we have production of human albumin in this disease mouse model. And so our previous data and our previous experiments have been terminated at around day 28 or day 30. And so this is now substantially longer. We have actually been able to take those experiments out beyond 63 days. And one of the interesting things now is, if you were to evaluate the area that is directly beneath the transplanted bioprinted therapeutic liver tissue, which is called the BTLT here, what you find is that the native mouse liver appears to contain many, many more healthy-looking hepatocytes than a sham implanted control. And so what you're able to identify in the treated animal are very clear cell borders, very few hepatocytes with large globules within them, And if you compare that to the area that is directly below a sham implant, that is just sutured and injured mouse liver, what you find are many hepatocytes. In fact, most hepatocytes are full of these globules, and the borders between the hepatocytes, because they are so hypertrophic and engorged, become very difficult to identify. So it appears that the implant here has had a bit of a trophic effect on the underlying mouse, and this is something that we're very interested in exploring and understanding mechanistically what may be going on here. So I think that, in summary, what we've been able to demonstrate in some of these early experiments is that implantation of these bioprinted hepatic tissues into immunocompromised mouse recipients results in very, very quick engraftment. We see very rapid evidence of graft perfusion. We're able to retain that original intended design and structure. And we are experimenting and exploring and optimizing that specific geometry and those specific designs. The grafts are well tolerated. I didn't present data today, but we have done, in fact, LFTs, and we've looked at liver panels in these mice. They show no signs of injury as a result of receiving implantations. And if we then look in the circulation, we're able to identify the presence of circulating human protein, albumin, alpha-1 antitrypsin, fibrinogen, and this bullet here says 28 days, but in fact, our more recent data now actually is out to greater than 90 days. So we have evidence of sustained function, and we have early indications that we have sort of a broad benefit. There is, for those who are interested, there is a potential mechanism through which we think this might be occurring, and we can certainly explore that offline. But it does appear that there is a direct effect in the area underneath the treatment of the recipient liver. We will be presenting this data and a little bit more, an extended amount of data at the AASLD meeting, so the Society of Liver Diseases, in Washington, D.C., October 20th to 24th. There will be a poster there describing those experiments in a little bit more detail, including, obviously, some more data that we're not quite yet able to share but are very excited about. So with that, I'd like to open things up to questions, and if you have any, please do submit.
Great, thank you, Ben. We'll now begin the Q&A segment. You can still submit questions through the questions pane in the attendee control panel. I'd like to point out we have received a lot of questions, so we'll try to get through as many as we can. If we can't get through them all, we promise to follow up with you offline. So let's go ahead and get started. First question from the audience. During the tissue generation process, cells are proliferating within the matrix? If so, what drives this proliferation, and does this have any impact on the tissue?
Yeah, so so that's actually a very, very good question, very insightful. So when we take cells and make what we would just consider our traditional cell banks from standard laboratory conditions, the cells are clearly proliferating, all of the cells except for the hepatocytes. So hepatocytes do not proliferate in that traditional sense in 2D culture, but the supporting cells, at least in this system, absolutely do. When you place them into a three-dimensional tissue arrangement, that growth seems to arrest, and that is not something that is specific to our system. So that is seen in many systems, including basic spheroids, right? So if one is to take, for example, cancer cells that grow, with pretty impressive kinetics, just in kind of the laboratory setting on plasticware, and place them into spheroids, by and large, that growth pattern slows down dramatically. What we anticipate happening in the therapeutic system, specifically in the liver, is that in response to liver injury, there are proliferative cues. So while we implant them in a quiescent state, that is to say that the cells are not proliferating within the tissue, when there is a regenerative response of the recipient, the host liver, we anticipate and expect that to be experienced at the level of the implanted tissue as well, and you would then expect to see graft expansion. And that is something that we are actively pursuing now and evaluating. So my answer would essentially be, in the normal setting, we see growth in 2D lab where cell culture conditions. When bioprinted, there is very limited growth. There's a very, very low turnover, both proliferation and death. And then in implants of non-injured animals, that low proliferation and death process continues. And we're now interested in understanding what stimuli might drive expansion.
Okay, thank you. We've got a couple questions here about kind of the cellular composition. So in the ExVive liver, are all the cells from the same donor? And kind of a related question, is it possible to create tissues from diseased human cells?
Yeah, so those are, yeah, spot on. So let's see. In the ExVive system, they are not all from the same donor. Some of the things that I didn't touch on today from a business perspective is that Organovo actually has a wholly owned subsidiary, Samsara Sciences, actually where we're broadcasting from today, that is focused on procuring donor tissue and isolating cells, both for use here, as scientists here at Organovo, but also to make commercially available to the community. And they are interested in procuring and isolating, purifying hepatocytes, as well as supporting non-parenchymal cells. So we have the ability to entertain making tissues from a donor-matched scenario. But typically speaking, in terms of quality control and understanding the growth kinetics, the behaviors, and all of the things that make for good product development, we tend to make master working cell banks regardless of whether or not they're donor matched in order to make our products. In terms of using diseased cells, the system is entirely amenable and open to that. And that is a fantastic suggestion in terms of looking at disease modeling. Can you take steatotic, cells from a steatotic liver? Can you take them from a NASH or a NAFLD liver? And do you then observe similar phenomena, whether that's at the protein, the gene expression level, or the tissue pathology level? That is something that we are acutely interested in.
Great, and I can further add to that. In the commercial offerings, we have worked with a number of clients to investigate different aspects of liver disease, either fibrosis and/or NASH. And currently, we offer commercially a way to induce a fibrotic phenotype with a variety of different fibrotic inducers. But we've also begun working with a few select clients in building disease tissue from the cell level upwards. And so we do that by sourcing cells via our subsidiary company, Samsara Sciences, in which they use pathologically graded donor livers. We can isolate those cell types out of those donor livers and then use those then to build disease tissues really from that cell level upwards. And so that's still a little bit early days, but is another exciting way in which we're looking to expand our disease modeling capability. Let's shift now and talk a little bit about some of the kind of clinical questions that I see here in the questions pane. So one big question is, when do you anticipate trying the liver patch in humans?
Yeah, so the $1,000,000 question. So what we have given guidance about is that is what we are trying to do in terms of IND filing. So we are marching down a path to anticipate filing in the next couple of years. So that's in probably calendar year 2020. By the end of that, we anticipate submitting an IND filing to the FDA. And I think that from that point forward, then obviously it's dependent a bit on establishing the trials and enrollment, but that is the anticipation. So you're looking at in the next few years here we hope to be prepared and ready to initiate the initial clinical trials.
So I guess a related question to that is, this is a unique therapeutic offering. It's not a drug in the classic sense of a small molecule. It's not quite a medical device. And so what thoughts do you have in terms of the unique regulatory pathway that would need to play out here in order to bring this to the clinic?
Yeah, so we're actually kind of looking forward to that process. So this is something that the agency, that the FDA has been really, I think, as forward thinking about as they can be. And so, this is obviously not a device. It is more akin to a biologic, but it's not really, it's not a gene therapy and it's not a traditional cell therapy. So we think that it fits really into the new efforts within the agency and within CBER to develop this OTAT, this Tissue and Advanced Therapies Office. And we think it fits right kind of into their purview, and they've expressed a lot of interest in bringing these types of therapies and these types of approaches to market ultimately. So we're kind of looking forward to working through that process, and we anticipate that that's the route that we'll be going.
So another question, I guess, is related to how you would scale this to humans. So one question is asking, how many patches would you use for a human, or potentially would you just use one patch but scale that to a large size? So maybe you could comment just in general about how you think about scaling this technology to human scale.
Yeah, so we hope that, we anticipate that some of the advantages to this type of additive manufacturing fabrication strategy, that at least one of the advantages there is the flexibility to make that on a patient-to-patient basis, right? So I think that fundamentally you'll have a product design that will be at its core the same across a variety of patients, but that there will probably be some specific anatomical drivers around what that implantation might look like for one patient versus another, and you may be able to fabricate something essentially to size, right? Fit to size. What has traditionally happened is that you have something large and it's then cut and trimmed. That may be something that's doable here, but we anticipate being able to, at least in the early days, with a limited number of patients in these kind of rare orphan diseases, we anticipate being able to look at that patient, make something that is appropriate, at least in terms of scale. And that has really been how we've approached the geometric and modular design aspects of the tissue development. So we think that both of those approaches, that is either multiple patches or one patch that's constructed specifically to that patient, we think that both of those approaches are probably viable.
Okay, one question here about just kind of the biophysical properties of the liver. How do you deliver oxygen to the internal cells of the 3D tissue is the question, and I guess I further might add, how does that maybe be similar or different in vitro cultured ExVive liver versus after implantation?
Yeah, so we, so what I can tell you is that nominally today, and I kind of perhaps glossed over this a little bit in the interest of time, the tissues at the time of fabrication, so what we would refer to as kind of the nominal thickness of those tissues, at least in these small animal mouse implants, are roughly 750 microns to perhaps a millimeter thick. There's a bit of remodeling that occurs as the tissue really kind of tightens and shrinks down and remodels a bit so that it's a bit thinner than that at the time of implantation. What we have found, at least in this system, is that the period of time that we keep those tissues in vitro, we do not see development of significant hypoxia within the core of the tissues. We do not see evidence of necrosis. When we implant them, we know from our histologic evaluation that perfusion is established in those tissues quite quickly, certainly before day 7. It's probably as early as day 3. And so there's a period of time, obviously, that is required for that kind of spontaneous microvascular anastomosis. But once that has occurred, obviously oxygen and nutrient both delivery and removal is occurring, through the vascular system as you would anticipate. So, as we go forward and think about scaling, we would like to keep things at a level that we can control that a bit, and then as you think about much larger tissues at the the adult patient size, perhaps you may need to think about some more sophisticated bioreactor systems, but that is something that we'll certainly be exploring in the coming years.
Okay, we probably have time for one more question, and you had shown in the PIZ mouse model that there appeared to be some improvement in terms of the underlying pathology in the host liver. And the question is, any comments on what you're currently thinking in terms of potential mechanism? Is it some sort of a secreted factor that's secreted from the bioprinted liver that then improves the pathology in the host liver, or is it perhaps some sort of a positive selection for more healthy mirroring hepatocytes?
Yeah, so the PIZ mouse is both attractive and a little bit complicated for a couple of different reasons. So it's attractive in that it is a nice model of, it is a model, I should say, of one of the liver diseases that we may be interested in treating. So it's slightly artificial in that you're overproducing this mutant form of the human alpha-1 antitrypsin, but the mouse has its own A1AT. That it is producing. So the mice do not develop a lung phenotype as you might see in the clinical scenario, but they do develop this liver pathology. And what is believed to occur naturally is that the hepatocytes, which can manage through autophagy that accumulated mutant protein that are kind of stuck in these globules, and these ER-bound globules, that those hepatocytes remain viable. And the hepatocytes that cannot, through essentially autophagy, cannot handle that polymerized vesicle-bound A1AT, that mutant A1AT, those hepatocytes then die. And that there may, in fact, over time, be selection for hepatocytes that can handle and process the mutant protein. We're wondering at this point if what we're actually seeing is through the implantation of healthy hepatocytes and kind of the supportive material that comes along with those hepatocytes in this bioprinted tissue. We're wondering, in fact, if we're improving the kinetics of that healing process. And so certainly the data today would support that hypothesis, but it's a difficult one to tease out specifically. So we think that there is quite a bit of evidence for this kind of global localized anyway effect, and we suspect that it's through the administration of healthy tissue to this diseased area.
Great. All right, that's all the time that we have for today's webinar. Please look out for our follow-up email with a link to view the recording on our website, organovo.com. As a reminder, Organovo will be presenting at the upcoming annual AASLD liver meeting in Washington, D.C., October 20 to 24th. If you plan to attend the conference, we hope you will stop by our poster sessions where we'll have several of our scientists, including Ben, presenting data on the therapeutic liver tissue as well as our commercial ExVive liver tissue. On behalf of Dr. Shepherd, myself, and the team here at Organovo, thank you for tuning in and have a great day.